Hey guys, welcome back. This is the me going over the progress check FRQ section for Unit 3, which is cellular energetics, which is AP Unit 3, which corresponds to chapters 8, 9, and 10 of our kind of Unit 2, biochem and cell biology. Um, so this question is the first of the FRQ questions of the unit progress check, and it looks like it deals with cellular respiration. So let's take a look at what it actually says. So researchers studied the relationship between glucose concentration, oxygen levels, and ATP production in one type of mammalian cells. Cells were isolated and cultured in growth medium containing either 1.5 millimolar glucose or 25 millimolar glucose, and at oxygen levels that varied from 0% to 21%. The researchers determined the concentration of ATP per cell under the different conditions. The ATP concentrations are shown as relative to the maximum ATP concentration obtained wherein cells were cultured in the presence of 25 millimolar glucose and 21% oxygen, standard culture conditions. The data are shown in Table 1. Okay, so let's take a look at the actual questions here. So for part A, you don't actually need to look at the data at all. All you need to do is describe the role of oxygen in cellular respiration. And luckily for you, we've spent a lot of time on that. Or spent a lot of time on it last semester. So we learned that O2 is going to be what we call the terminal electron acceptor. So what that means is electrons are going to get knocked off as glucose is oxidized in the process of glycolysis and the Krebs cycle, also called the citric acid cycle. And if you remember, those electrons get picked up by electron carriers like NADH and FADH2. And remember we talked about those being like Lyft and Uber, and they pick up electrons and they bring it over to the mitochondria and they drop them off in the electron transport chain of the ETC, or electron transport chain. And what happens is that oxygen is very electronegative and it sort of pulls the electrons down through the electron transport chain, and they release potential energy. They release energy every time they move down towards oxygen. And at the very end, they're very low energy and they bind with oxygen and a proton to produce water. But that energy is used to create what we call a proton gradient, when that energy is used to move protons into the intermembrane space of the mitochondria. And then when those protons come back, the only place that they go back is through a protein that we call ATP synthase, because it's an enzyme that actually produces ATP. Okay. Hopefully this is ringing some bells, because we spent a good 2 or 3 weeks on this in October or November probably. Okay, so let's take a look here. So for part B, construct an appropriately labeled graph to represent the data in Table 1. Okay, so let's take a look at the experiment, see what the data are showing us. So looks to me like we have 2 experimental groups. We have one that was done in the 1.5 millimolar glucose condition, the other in 25 millimeter glucose conditions. So it looks like we got really 2 sets of data here that are both being compared, to the same variables, right? So percent of oxygen versus relative concentration of ATP per cell. So there's a couple of different ways you could probably represent this data, but for our purposes, I'm just going to say, okay, this reminds me of the cellular respiration experiment that we did, except we were measuring oxygen consumption instead of ATP. But you still had, 1 or 2 sort of experimental groups that you were taking data on. So it doesn't really matter what you put where. I put percent of oxygen, I think. How did I do it? Yeah, I put percent of oxygen here on the X or horizontal axis, relative concentration of ATP per cell up here. I really think it matters which one you do. And so what I did is I chose to make a line graph, and I just said, okay, I'm going to have blue represent my 1.5 millimolar, my black represents some 25 millimolar. You could do it solid dashed, however you want to do it is fine. And then you just kind of apply your points, right? And then I just sort of connected the dots here. Um, and again, I made sure that my graph was labeled with, my axes were labeled and my graph was scaled out appropriately. So let's see what they were looking for. So oxygen is the terminal electron acceptor of electrons that pass through the electron transport chain. Great, we covered that in our answer. And then here, again, there were a couple different ways you could have done this. And so let me just go back here for a second, because I'm sure some of you, maybe if you've tried this before, which hopefully you have, and are just kind of checking your answers here, maybe some of you made a bar graph and said, okay, I'm just going to make, one bar for 1.5 millimolar. The second bar is going to be 25 millimolar. And you had two bars that were, one was like that, and then the other one was up here like that. And this one was, slightly higher. You could do it that way. I don't think it's better than what I did. I think both of them are fine. Because it says the sketch bars curve meet all of the criteria below. So that leads me to believe that you could make a bar graph and it would be fine. It does say correctly plotted line graph here, but I feel like maybe this should say data, correctly plotted data. Because if it says that the bar graph is okay, then I don't see why they would be taking off points here for not doing a line graph. But either way. Let's look at part C. So describe the relationship between the concentration of glucose in the culture medium and the ATP concentration in the cells. I mean, it doesn't look like it made a whole lot of difference just eyeballing it. Really, if I was going to do this, if I was going to write a question like this, what I would do is I would, without a doubt, throw in some standard error. Just because just eyeballing it is, I don't know, it's kind of insufficient to really get a good answer. But if you had something like that, what you could see is these error bars overlapping, which would mean that there is no significant difference between the concentration of glucose and the ATP concentration. They're just really too close to call. So again, without those error bars, you can't really say any more than, small difference or insignificant difference. Basically not a huge impact. Okay, so for part D, I left myself a good amount of space here because this is actually probably one of the harder ones that I've seen just in the progress checks so far. Okay, so I'm going to draw that little line, and I'm going to make this my inner mitochondrial membrane, and you'll see why here in a second. Okay, so what this question is asking is, in a further experiment, the researchers add a compound to the cell growth medium that both binds and releases protons and hydrogen ions, and also passes through lipid membranes. Predict the effect of this added compound on ATP production by the cells. So justify your prediction. So, okay. So what we have here is we've got our inner mitochondrial membrane, and this is a lipid membrane. It's the same as ours, right? It's a phospholipid bilayer. So when it says it can pass lipid membranes, the lipid membrane that we're talking about here is going to be the inner mitochondrial membrane. And if you remember from part A, what we had decided was that there were hydrogen ions at play because the energy released by moving those O2s down the electron transport chain is basically used to move hydrogen ions into the inner membrane space, right? So let's just get a quick idea of where we are here. And there might be a few down here, but most of them are going to be up here because these are being pumped out and into the inner membrane space. So again, let's just label our geography here. So this is going to be the inner membrane space. And this is going to be our mitochondrial matrix down here. Okay, so as you guys saw, so if this is the electron transport chain, this general, this general area over here might have some, might have a couple of proteins here, and what that's doing is that's moving H plus in, right? So we're going to label this our electron transport chain, okay, and that's moving hydrogen ions in there. And then you've got this other protein, okay, which is going to be our ATP synthase, and that's going to be responsible for letting H plus go back down its concentration gradient. And when it does that, that is going to basically catalyze the phosphorylation of ADP into ATP. So we're going to produce ATP that way. So basically, this is the situation that you're kind of supposed to have in your head when you're trying to answer this question. So basically, what I see here is say, okay, you're adding a compound that both binds and releases proteins and is going to pass through the lipid membrane. And really what I think they're talking about is, okay, if it binds these and then it goes, it's going to pick up more protons, more hydrogen ions, where there's more, right? So it's going to pick up a bunch of these, right? And then it's going to kind of diffuse down its concentration gradient, and because it could pass the membrane. And then it's going to release hydrogen ions into the mitochondrial matrix, right? So this here, right, the thing that I'm circling, right, this is our, mystery compound, whatever it is. Okay, so there's our compound that can take hydrogen ions across the lipid membrane. Okay, so if this happens, that's going to decrease the concentration gradient, right? It's going to decrease the proton gradient. So less protons are going to go through ATP synthase, and that's going to decrease the ATP production. And so our prediction would be, decrease ATP production. And then the effect of this, let's see, so that's our effect, is that it's going to decrease ATP production. And we're going to justify it by saying that, it decreases the proton gradient if it picks up hydrogen ions or protons in the inner membrane space and moves them into the mitochondrial matrix without having them pass through ATP synthase. Okay, so again, let's see what they're looking for here. Okay. So, sorry. Here we go. Okay, so it seems to me like they are looking for little to no effect on the ATP concentration of cells for part C. Part D, amount of ATP produced will decrease or no ATP will be produced. And then, okay, importantly, here it says that more likely to bind protons where protons are in greater concentration in the intermembrane space. Also indicates that if the compound then carries the protons across the inner membrane to the matrix, there will no longer be a proton concentration slash pH slash electrochemical gradient across the inner membrane, and protons will not flow through and power the ATP synthase enzymes. So yeah, it's kind of a more difficult one for sure, but it's definitely doable. Like I said, I mean this is something that you've looked at before. So if you kind of have this in your head, and again, don't be afraid to sketch things out, right? Um, you would conceivably be able to come through and answer that question correctly. Now, I would recommend that something like this that's really involved, if you're really concerned about time, maybe save something like this through the end, right? Because as you've seen coming through, there's a lot of questions here in these progress checks that are easier or less time consuming to answer than this. So maybe this is an example of something that you would move through and you can skip initially and then come back to. Okay, so we'll take a look at the second FRQ question in this progress check in just a second here. Okay, thanks a lot.